Okay, so we are done with these three structures. Now the one more biological molecule which is present in your body is known as DNA, right? And the information about the DNA are is, let's go through it. Full abbreviation of the DNA is deoxyribonucleic acid. And where can you find this DNA? In chromosomes. If it is eukaryotic cell, then in the nucleus, and if it is a prokaryotic cell, you will find it in the cytoplasm. Okay? Again, DNA, deoxyribonucleic acid. If it is a eukaryotic cell, you will find it in the chromosomes inside the nucleus. And if it is a prokaryote, you will find it in the cytoplasm floating. Okay? In the chromosome, single loop structure. Remember chapter number 1, we have discussed that how the bacteria is different from the animal in the plant cell. Right? One of the part was it was prokaryote, so it does not have a nucleus, but it does contain a chromosome, which is a single loop of chromosome and present in the cytoplasm. Okay? Okay. And DNA is made up of the building block known as nucleotides, right? Okay. What are nucleotides then? ठीक है, अभी हम उसको पढ़ते हैं. The element makeup. What are the elements which are necessary for making the DNA? Is carbon, hydrogen, oxygen, nitrogen, and phosphorus. ठीक है? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay. And what is the structure of eukaryotic DNA? It's supposed to be two-stranded wound around each other called double helix. See? This is how it looks like. Double helix. And it's wound around each other. Helix का मतलब है कि it's twisted. ठीक है? So there are two strands. You can see there are two strands and they are twisted around each other. They are wound around each other. That's why it's known as double helix. Okay? It's double helix. Okay. And between these double helix, can you see these structures present which are holding these two strands? They are the nucleotides. Okay, they are the one nucleotide. And nucleotide actually make up a sequence of a code. Okay, there are four types of nucleotide you can find that is known as, or four type of bases you may find that is known as A, C, T, G. Examiner says that you don't have to remember the full name. So let's go to A, T, C, G. Okay, structure of the nucleotide. How the nucleotide, what is actually nucleotide? This is the base. How many bases are present? Four bases are present. What are those? A, C, T, G. Those four bases, when they are attached with the phosphate group and the deoxyribose sugar, ठीक है? Nucleotide. अब हम कह रहे हैं कि this is the building block of DNA. That's nucleotide. What is actually a nucleotide? ठीक है? This is a nucleotide. ये जो आपको एक नजर आ रहा है ना यहाँ पे ब्लू कलर का, ग्रीन कलर का, रेड और पर्पल, दिस एक्चुअली वन मॉलिक्यूल लुक्स लाइक दिस. ठीक है? वन बेस अटैच विद द शुगर एंड विद द फॉस्फेट ग्रुप. ठीक है? कौन सी शुगर? डीऑक्सीराइबोस. जो उसका नाम है ना, डीऑक्सीराइबोस न्यूक्लिक एसिड, उसी नाम एक्चुअली वुड हाफ नेम इज द शुगर, डीऑक्सीराइबोस शुगर. ठीक है? And it's bonded with a structure known as phosphate. So phosphate, sugar, कौन सी sugar? Deoxyribose nucleic acid, deoxyribose sugar. And on one end of the sugar, you will find, this is a sugar, right? On one end, you will find phosphate. And on the other end, you will find a base. Which base? There are four bases. T, A, T, C, G. ठीक है? One nucleotide would contain one base. So there, how many nucleotides are there now? Four. How many bases are there? Four. ठीक है? And they are actually joined together. They are in a pair form actually. ठीक है? When they are on two strands, they who will face who? दोनों strands पे हैं. इस पे भी आपको कुछ नजर आ रहे हैं, देखो. और यहाँ पे भी आपको कुछ लगे हुए नजर आ रहे हैं. तो इनके
So like
ऑपोजिट में कौन है?
A will face T and C will face G.
Sorry.
They're pairs, right? So like A will face T, right?
Yes. A will face T
And C will
and
face.
C will face G. ठीक है? Separately, how many bases there are? There are four bases, but when they are a pair form, the A will be paired with T and C will be paired with G. ठीक है. This is how. And when they are bound with each other, ठीक है, on the strand, this gives it actually a double helix structure. What are these nucleotides are forming? ये बना क्या रहे हैं? They are actually making a sequence, a code. हम कहते हैं ना code of life. ठीक है. If I'm saying you have genes, your hair color is black, your eye color is black, ठीक है, your skin color is wheatish, you are tall. What is these? These are the characteristics. Characteristics. How did you get these characteristics? Which are enclosed in genes. And what are the genes? Genes are these nucleotides. It's a sequence, actually. शॉर्ट सीक्वेंस, ये एक सीक्वेंस है. मे बी दिस, ये जो सीक्वेंस है आपके पास 1 2 3 4 5 6 न्यूक्लियोटाइड का जो सीक्वेंस है, ये एक जीन होगा, ये कोई स्पेसिफिक काम करेगा. फिर इसी तरीके से नीचे एक और कोई जीन होगा, वो कोई और काम करेगा. सो देयर आर लाइक मिलियंस ऑफ जीन्स ऑन द डीएनए. डीएनए बहुत बड़ा होता है. ठीक है. इट्स लाइक अ डेटाबेस. इफ यू नो द कंप्यूटर, इट्स लाइक अ डेटाबेस. पूरी बॉडी का डेटाबेस जो है वो आपके डीएनए के अंदर होता है. कैसे उठते हो, कैसे बैठते हो, कैसे खाते हो, वॉट एवर यू हैव in your body, what type of enzymes you have, what type of antibodies you have, how lean you are, ठीक है, how chubby you are, ये हर चीज आपकी बॉडी के अंदर कौन कंट्रोल कर रहा है? these DNA. DNA contain these codes, this sequence in form of genes. They are controlling everything. ठीक है. सो इट्स अ कोड ऑफ लाइफ, यू कैन से दैट. ठीक है. तो ये आपका डीएनए हो गया. तो डीएनए के हमने क्या पॉइंट्स पढ़े? नंबर 1, full abbreviation of the DNA, deoxyribonucleic acid. Where can you find it? In eukaryotic cell, you will find it in the chromosome, in the nucleus. And if it's a prokaryote cell, you will find it in the chromosome, but in a single loop form in the cytoplasm. And how they are made up of? The building block of the DNA is nucleotides. What are the elements which make up the DNA? Means this nucleotide किसका बना हुआ है? ये कौन से elements हैं? You will find carbon, hydrogen, oxygen, nitrogen, and phosphorus. How does the eukaryotic DNA look like? Double helix. Two strands wound around each other in form of double helix. ठीक है. And what is nucleotide? If I'm saying that my DNA is made up of nucleotide, what is actually a nucleotide? It's actually a base, any base which which is attached with a sugar and a phosphate group. ठीक है. This is known as nucleotide. So there are actually 4 bases which is attached with a nucleotide. If it's A, it will be A nucleotide. If it's T, it will be T nucleotide. If it's C, it will be C nucleotide. If it's G, it will be G nucleotide. ठीक है. And they are actually, they do exist in a complementary form or they exist in a pair form. So A pair with T, C pair with G. ठीक है. Okay, ji. So this is actually about the DNA and we are done with the chapter. This is done. Okay, let's start the new chapter. That is enzymes. Enzymes. Yeah. Okay. Enzymes, what you need to know in the enzymes, what is catalyst, right? And what is biological catalyst? What is the definition of enzyme? What is the definition of catalyst? And what is the structure of the enzyme? How do they work? What are the structures bind with the enzyme? And what factors affect the enzyme? And what happens if the factors changes? What will be the enzyme activity? ठीक है. Enzyme आपके पास explanation. These are the actually key points you need to remember. So what is catalyst? Enzyme, a catalyst is something which speeds up the reaction, right? Speeds up the reaction, speeds up the chemical reaction without being used themselves or without being used up. Means they would come, they would speed up the reaction, ठीक है? but they will not be used up in that reaction. They will come, make the work easy for you, and they will go away. That is actually known as catalyst, right? Catalyst something which speeds up the reaction, and you can find some catalysts which are organic or inorganic. But some catalysts which you may find, this temperature is a catalyst, speeds up the reaction. Fuel is a catalyst, speeds up the reaction. Okay? But if you find these catalysts in the biological or in the living organisms, right? In the living organisms. Now, these catalysts will now known as enzyme. So enzymes are actually the biological catalyst. When I'm using the key term biological, what do you mean by biological? Means they are present in the living organisms. Catalyst means speeding up the reaction. So what are enzymes? They are the catalysts which are found in living organisms. But those who are not found in the living, which are present outside the living organism, they are known as catalyst. This iron, you have studied the periodic table. In the periodic table, there's a transition elements. Some of the transition elements are catalyst. Vanadium is a catalyst. Iron is a catalyst. ठीक है? लेकिन एंजाइम किसे बोलेंगे? वो कौन से कैटलिस्ट होते जो एंजाइम होते which are found in the living organisms. That's why the key term, the first key term for the definition of enzyme is, what is enzyme? Enzyme are the biological catalyst which are made up of protein, and they speed up the chemical reaction without being used themselves, right? ये definition हो गई enzyme की. All are the key points. Now, in this red box you have seen, I have mentioned enzyme will give the Buret test. What if I want to test the enzyme for the food test? I want to know, see which biological molecule the enzyme is made up of. Okay, so which test I would do? Buret test. Why? Because it is made up of proteins, so Buret test will give me the positive test. Okay, so enzymes are made up of proteins. And which test will we perform? That is the Buret test. Okay, I hope this is clear to you. And enzymes are present in our body, and whom they are working on. The substance the enzyme are working on, or substance they are actually speeding up the reaction for, are known as substrate. Okay, substrate are the substances on which the enzymes act on. अब ये कौन से substrate है? ये जो भी आपने पढ़ा है biological molecules, carbohydrates, proteins, fats, they are all substrates. I want to break down starch. Amylase is the enzyme. Starch is the carbohydrate. ठीक है, biological molecule, carbohydrate, you will get it from the food. तो वो क्या हो गया? वो substrate हो गया. ठीक है? Okay. If you want to skip this, you can. ठीक है? अभी मैं ये इतनी डिटेल आपके पास नहीं रही. एक्चुअली क्या होता है कि एंजाइम रिएक्शन जो है ना, वो उसकी स्पीड जो है वो फास्ट हो जाती है. यानी रिएक्शन, इनिशियल रिएक्शन जिसे हम कहते हैं ना, इनिशियल रिएक्शन वुड बी लोअर डाउन. ठीक है, यानी कि पहले अगर आपका एक्सपेरिमेंट 80 पे स्टार्ट हो रहा था, एंजाइम क्या करता है? वो आपका एक्सपेरिमेंट 20 पे ले जाता है. कम एनर्जी चाहिए हुई आपको. कहाँ आप 80 पे स्टार्ट कर रहे थे, कहाँ आप 20 पे स्टार्ट कर रहे थे, उसको हम एक्टिवेशन एनर्जी कहते हैं. सो एंजाइम्स एक्चुअली लोअर द एक्टिवेशन एनर्जी. ठीक है, यानी कि जो काम आप बहुत हाई टेम्परेचर से कर रहे हो, अब वही काम आपका लो टेम्परेचर से हो रहा है, तो आपको ज्यादा एनर्जी नहीं देनी पड़ रही. दिस इज द बेनिफिट ऑफ हैविंग एंजाइम. Enzyme are actually making my work easy. First of all, speeding up the reaction, and secondly, they are lowering the activation energy. So my work is becoming faster. ठीक है? If you want to skip this part, you can. अपनी slide में से हटा देना से. Now, uses of enzymes, major part. They may ask you, list some uses of enzymes. There are 7. You can learn any of them, and they are being taken from your syllabus. कुछ outside का नहीं है, सब आपके syllabus से लिया गया है. Digestion, cellular respiration, you have studied that. Germination of seed, अभी नहीं पढ़ा, लेकिन of course utilized in germination. Photosynthesis, you have studied photosynthesis. Carbon dioxide, how do you think the glucose is formed? When the carbon dioxide and the water molecules are combined together. Who is combining them together? खुद ही एक दूसरे के साथ मर गए. Actually, those enzymes which are present in the chloroplast are combining them together. So here is the use of enzyme. And in the mitochondria, respiration is occurring. What happening in the respiration? The glucose और ऑक्सीजन जो था वो आपस में बाइंड कैसे कर रहा है? ये एंजाइम्स ही तो हैं जो इन दोनों को बाइंड करवा रहा है, जिसकी वजह से उसने ग्लूकोज को ब्रेक डाउन कर दिया, उसमें से एनर्जी निकाली. ठीक है. And formation of the large proteins from amino acids. Remember, um, nitrates, when they bind with glucose, they form proteins and amino acid. वहाँ पे भी enzymes चाहिए होते हैं आपको. ठीक है. And pectinase is a part of 11th grade. Lactase enzyme is a part of 11th grade. ठीक है. If you want to skip these two, you can. अब आते हैं हम कैरेक्टरिस्टिक ऑफ एंजाइम्स. ठीक है. What are the characteristics of enzymes? What are the features of enzyme? Number one, it should speed up the chemical reaction. Number two, it should remain unchanged in the chemical reaction. ठीक है. वो reuse होता है, used up नहीं होता. ठीक है. इसकी ये प्रॉपर्टी है एंजाइम की. आते हैं, काम करते हैं. ठीक है. एक सबस्ट्रेट पे काम किया, फिर उसने दूसरे सबस्ट्रेट पे काम किया, फिर उसने तीसरे सबस्ट्रेट पे काम किया. खुद उसको कुछ नहीं हो रहा, वो सबस्ट्रेट के ऊपर एक्ट कर रहा है. And they can be reused over and over again. And if something which is being reused again and again, I would be needing in a minute amount. Why would I be needing in a huge amount? जो वो मेरा काम थोड़ी सी चीज में हो रहा है, तो मुझे ज्यादा चीज की क्यों जरूरत पड़ेगी उसे. ठीक है. Okay. And they have a place, a specific place where that substrate bind. How the enzyme actually works? Enzyme has a specific space, a specific place or area in their structure where that substrate binds, and it's supposed to be complementary. Complementary means it should be a match. This area where the substrate binds is known as active site. Enzyme recognize the substrate when they attach at the active site or binds at the active site. ठीक है. एंजाइम है, सबस्ट्रेट आया, उस एक्टिव साइट में फिट इन हो गया, तो एंजाइम काम करेगा. अगर उस शेप से कॉम्प्लीमेंट्री था तो एंजाइम काम करेगा. शेप डिफरेंट है तो एंजाइम काम नहीं करेगा, सबस्ट्रेट काम नहीं करेगा, कोई और सबस्ट्रेट होगा उसका. सो व्हाट इज द की टर्म for the active site and substrate actually fitting in the active site? It should be complementary. Complementary means it should be a matching. ठीक है. Okay. So, acha, please remember that some students think that enzymes only break down the substances. No, they also make up, like photosynthesis. Glucose is being made up by combining carbon dioxide and water with the help of enzyme. In the respiration, the glucose molecule is broken down with the help of enzyme into carbon dioxide and water. So enzymes are helpful not only in breaking down the substances, they also make up the substances. Amino acids are present. I want to make a large molecule of a protein. What I would do, I need to join them up. So here would be enzyme which will help them to join them up. ठीक है. I hope this point is very clear to you that enzymes not only break down, they also build up. They do both. What they are doing, they are only speeding up the reaction. Whether it's breaking down or build up, it's speeding it up. ठीक है जी. Okay. And how would you recognize if enzyme? There is a prefix as ase, जैसे amylase, maltase, lipase, fructase, sucrase. ठीक है. Okay. And enzymes are protein based. These are some characteristics of enzymes. All right. Okay, ji. Active sites, what are active sites? I have just explained to you, is a region in the enzyme where the complementary substrate fits in. And then they will form enzyme substrate complex. It's very necessary for the function. The complex needs to be made. अगर complex नहीं बनेगा तो काम नहीं होगा. When the substrate fits in at the active site, that will form enzyme substrate complex. ठीक है? Okay. Now, how do they work? There is a theory for their function. How do they work? Their theory is known as lock and key, a theory, or you can say this is a hypothesis that's known as lock and key hypothesis. जैसे यहाँ पे, if I'm saying that substrate supposed to be complementary, ठीक है, तो ही काम करेगा ना सबस्ट्रेट, वरना तो काम नहीं कर सकता. एंजाइम कैसे काम करेगा? कोई भी स्ट्रक्चर आके उसमें लग गया तो नहीं पॉसिबल है ना, सबस्ट्रेट का एक स्पेसिफिक शेप होना चाहिए, जस्ट लाइक अ लॉक इन की. Do you think any key can open the lock? No. You need to have a specific shape of the key, a specific shape of the key which can fit in the lock. Then the lock will be opened. ठीक है? We are using this phenomena in a process, in a hypothesis known as lock and key hypothesis. This lock and key hypothesis is actually telling you how the enzyme and substrate works. ठीक है? तो इसका स्टेटमेंट क्या है? ये जो ये फर्स्ट आपको पॉइंट नजर आ रहा है ना, ये एग्जैक्ट स्टेटमेंट है. एक्सप्लेन लॉक इन की हाइपोथेसिस, यू कैन राइट दिस डाउन. ठीक है? डिस्क्राइब लॉक इन की हाइपोथेसिस, यू कैन राइट दिस डाउन. स्टेट लॉक इन की हाइपोथेसिस, यू कैन राइट दिस डाउन. ठीक है? So when the substrate molecule fits in, you can see this is the underline key point. An enzyme at their active site, they form enzyme substrate complex. Then the enzyme converts substrate into product without being used themselves chemically, and they can start the new reaction. This is lock and key hypothesis. And lock is actually enzyme and key is the substrate. जैसे मेरी बॉडी में एंजाइम्स हैं, तो मेरी बॉडी के पास कौन आ रहा है? सबस्ट्रेट आ रहा है कि एंजाइम आ रहा है? सबस्ट्रेट आ रहा है ना. वट एवर आई एम ईटिंग द फूड, दैट इज द सबस्ट्रेट, एंजाइम विच आई ऑलरेडी हैव. ठीक है? सिमिलरली लॉक इन की में भी अगर आप देखो तो लॉक तो किसी के पास चल के नहीं आता ना, की आती है लॉक के पास. दैट्स व्हाई की इज द सबस्ट्रेट एंड लॉक इज द एंजाइम. ठीक है? सो फॉर एग्जाम्पल देयर आर डिफरेंट enzymes, ठीक है, and substrates, like for example maltase. Maltase is a key or a lock? Maltase is a lock. Lock, very good, it's a lock, right? Because it's an enzyme. और इसका substrate क्या हो जाएगा? maltose. And product क्या हो जाएगा उसका? The product would be glucose. तो आपको ये याद करना पड़ेगा, what are enzymes and what are their substrates and what will be the product. Because the next chapter we are moving to, that is the digestion, वहां पे ये सारी चीजें conclude हो रही है. ये सब वहां पर आ गया. All those biological molecules and what are the enzyme substrate, how do they work, we are going to use this phenomena in the next chapter. ठीक है? Okay. Now, we are done with this. Last part of this chapter, factors affecting the enzyme activity. Two factors very important. One is the temperature and other is the pH. Okay, how the temperature affects the enzyme activity. As the temperature increases, okay, so what is explanation here actually? When we increase the temperature, the kinetic energy of the enzyme and the substrate molecules increases as well. Okay? Which leads to collision. More enzyme and substrate collides, more enzyme and substrate complex will form. अगर हम बोल रहे थे यहाँ पे कि एंजाइम सबस्ट्रेट कॉम्प्लेक्स बनना जरूरी होता है रिएक्शन के लिए, अदरवाइज एंजाइम कैन नॉट वर्क, दे नीड टू कम क्लोजर ना. हाउ वुड दे कम क्लोजर? दे नीड टू कोलाइड विद ईच अदर. ठीक है? सो temperature is increasing their kinetic energy so they can collide. More they collide, more the enzyme substrate complex will occur and more the products will form. Faster will be the reaction. Okay? So this is actually how the substrate help in the how the temperature helps in the increasing the enzyme activity. Okay. Okay. Now, there's a key term, optimum temperature. It means that there will always a point for all the enzymes where they work best. So the key point for working best is optimum. All right? Okay. But what if the temperature exceeds? What is kept on going? The enzymes will denature as the active sites will change the shape. Very nice. They will lose their active sites. Yes, very good. You can see They are losing the active site. Now they are losing the complementary shape with the substrate. How the substrate is going to attach with the or fits in with the enzyme. This now enzyme is useless for us. So this is now known as denaturation. Denature हो गया enzyme हमारा. And this phenomena is known as denaturation. ठीक है. So अगर definition आती है आपके पास, take the term denaturation, it's written here. When enzyme loses their active site and change their shape due to high temperature or extreme pH, that is known as denaturation. ठीक है. अब यहाँ पे आपके पास क्वेश्चन आता है, describe the temperature, describe how the temperature affect the enzyme activity and explain how the temperature affect the enzyme activity. दो सवाल आ रहे हैं. Describe and explain. कैसे बताओगे? You have a graph. इसमें ग्राफ आता है आपके पास. ठीक है. आप ग्राफ देख के बताओगे कि क्या हो रहा है यहाँ पे. If I'm saying कि describe the graph. ठीक है. Describe how the temperature is affecting the enzyme activity. The key term is describe. ठीक है. पहले हम describe कर रहे हैं. For the describe, ठीक है, I just need to use the values which are written in the graph. मुझे सिर्फ values को use करना है. ठीक है. कैसे? How would I create my answer or form my answer? At 0, enzymes were inactive. Please remember 0 के ऊपर enzyme denature नहीं होता, inactive हो जाता है. ठीक है. As the temperature increases at 10, ठीक है, the enzyme activity is also increasing. At 20, kept on increasing till 40, where it is at its peak. देखो मैं कोई की टर्म यूज नहीं कर रही, मैंने कोई काइनेटिक एनर्जी का वर्ड यूज नहीं किया, मैंने कोई ऑप्टिमम का वर्ड यूज नहीं किया. ठीक है. कोई कोलिजन का वर्ड यूज नहीं किया, मैं सिर्फ डिस्क्राइब कर. डिस्क्राइब का मतलब व्हाट यू सी व्हाट इज हैपनिंग. जो दिख रहा है, क्या दिख रहा है मुझे? वैल्यूज दिख रही हैं बस यहाँ पे. दैट्स इट. मुझे नहीं पता यहाँ काइनेटिक एनर्जी थी कि नहीं थी. कहाँ दिख रहा है? मुझे तो नहीं दिख रहा. When I will explain it, that is now beyond this value. अब हो कैसे रहा है? Explain means why it is happening, how it is happening. ठीक है. This, if you take care of these command terms, then there no one would be there to say कि आपका आंसर incorrect था. ठीक है. These command terms.
एक्सप्लेनेशन में हम सारा लिखेंगे कि इसमें ग्राफ में हो रहा क्या and describe you just have to use the values given.
The values, and in the explanation
Okay.
you will use the value, but you will tell what is happening, right? For example, describe, for the describe, at 0 the enzymes were inactive. ठीक है. As the temperature increased, ठीक है, at every 10 degrees centigrade, ठीक है, after every 10 degrees centigrade the enzyme activity is increasing, ठीक है, till it reached to its peak at 40. As the temperature increased from 40, the enzymes have started to lose its enzyme activity, and the enzymes and the क्या कहते हैं उसको, the reaction stopped. किस पे? 60 पे. यहाँ पे लगाओ, for example it would be 60 to 63. The reaction stopped at 60 to 63. This is my description. ठीक है. Now when I'm explaining it, ठीक है, अब मैंने क्या चीज यूज किया? I have used the command term that is explain. For explain, now you will use the key points. ये key terms. अब key terms use करोगे using the data. अब कैसे key terms आप use करोगे? At 0 the enzyme activity was inactive. As the temperature increasing, ठीक है, after every 10 degrees centigrade the temperature is increasing, the enzyme activity is also increasing as the kinetic energy is increasing. अब क्या key term आ गया यहाँ पे? Kinetic energy. ठीक है. Kinetic काइनेटिक एनर्जी. ठीक है. काइनेटिक एनर्जी इंक्रीज, तो क्या इंक्रीज हुआ? कोलिजन इंक्रीज. किसकी कोलिजन? एंजाइम. मोर, व्हाट विल हैपन देन? मोर एंजाइम substrate complex will form, so there will be more product. ठीक है, ये आपका क्या हो गया, यहाँ पे एक्सप्लेनेशन हो गई। ठीक है, at 0 enzyme activity was inactive
Could you
as the
explain
temperature
the explanation
increased.
part again?
Hmm, sorry.
I was saying, could you explain the explanation part again?
Huh, okay. In the explanation you have to use the key terms, using the values as well, दोनों को बताओगे क्योंकि वो पूछ रहा है ना आपसे कि हर वैल्यू पे क्या एक्सप्लेनेशन हुई. So at 0 it was inactive, enzyme activities were inactive as the temperature started to increase. ठीक है, at 10 the kinetic energy due to the temperature of the enzyme substrate also increased, which causes the collision and substrate enzyme complex were formed. You know, more enzyme substrate complex will form, more product will be formed, more enzyme activity will be occurring. Okay? So as the temperature increases, the kinetic energy of the enzyme substrate molecules also increases. Okay? Till it reaches to 40. विच वॉज द ऑप्टिमम टेम्परेचर. अब यहाँ की टर्म यूज हो गया ऑप्टिमम, पहले हमने पीक बोला था, अब हम ऑप्टिमम बोले क्योंकि ये इसका की पॉइंट है यहाँ पे. ठीक है, टिल इट रीच टू इट्स ऑप्टिमम टेम्परेचर दैट इज 40. एज द टेम्परेचर इंक्रीज्ड the enzyme starts to lose or starts to become denatured till the reaction stopped at 62 when the enzyme were completely denatured. Completely denatured वो 62 पे हो गया. My reaction has stopped now. ठीक है, see how I have used the values and the key points or key terms here in the explanation. For example, अगर question आता describe and explain the graph, फिर क्या लिखते? Describe and explain, both key terms are there.
So we would like use the values and the key terms
Yes, you will use the
together.
values and the key terms both. ठीक है, दोनों बताओगे आप यहाँ पे. सही है. So this is how the examiner test you. आपको ऐसे टेस्ट करता है, वो आपको क्लू दे रहा है कि मुझे आंसर में चाहिए क्या. आप क्लू देख के बताओगे. अगर आपको क्लू पता होगा तो आप सही बताओगे. दिस इज व्हाट दीज जिसको कहते हैं, दीज आर द टेक्निकल स्किल्स ऑफ द पेपर. Knowing the content is not the skill. How to portray the answer is the skill in the paper. ठीक है, this is the point where the students lose their marks. They know the content, they don't know how to portray the answer. They do not understand what the examiner is asking them. वो क्या पूछना क्या चाह रहा है वो. ठीक है, वो कहना क्या चाह रहा है समझ ही नहीं आती बच्चों को. क्यों? क्योंकि पास्ट पेपर सॉल्व नहीं कर. The best way to understand what the examiner is saying or how to go into the mind of the examiner is best way to solve the past papers. Solve the past papers. ठीक है, past papers actually tells you how the examiner thinks and how you are going to be graded. कैसे ग्रेड करेंगे, how would I am going to grade. हर टॉपिक का की टर्म होते हैं. जैसे इस टॉपिक की की टर्म्स है आपके पास. ठीक है, increasing kinetic energy, collision, substrate and complex molecule, denaturation, losing of the active site. ये क्या है, ये इसकी की पॉइंट्स है. तो remember, learn the topic through key points. ठीक है, do not learn it as the paragraphs के पैराग्राफ रट लिया आपने. नहीं, की पॉइंट्स उठाओ. की पॉइंट से याद करोगे, देखना आपको ज्यादा इफेक्टिवली याद होगा. ठीक है, ओके, दिस इज ऑल अबाउट temperature. ठीक है, this is another graph. You can go through it later on. pH is another factor. pH में क्या है कि इसमें slowly gradually नहीं increase होता. जैसे आप यहाँ पे देखो in the temperature graph, it was starting from 0, slowly gradually it was increasing. In the pH actually what happens, it abruptly increases. ठीक है, or starts or it becomes activated whenever the enzyme achieve their optimum pH, they are activated all of a sudden. ठीक है, they will work. ठीक है, as the temperature starts to, as the pH starts to change from their optimum pH, they will deactivate it again. ठीक है, जैसे उसको pH मिलेगा काम करेंगे, pH खत्म काम खत्म. तो इसका ग्राफ में थोड़ा सा चेंज आप देखोगे, there is no gradual increase, there is an abrupt increase. ठीक है, an abrupt fall, abrupt increase and abrupt fall. ठीक है, okay. Now is increase or decrease from the it. इसके पास भी क्या है, the best pH on which the enzyme work is the optimum pH. right? What you need to remember, pH in different human elementary canal. अभी हम जो डाइजेशन पढ़ेंगे उसी से ही पूरा कनेक्टेड है. मैं यही एक्सप्लेन कर रही हूँ तो वहां मुझे एक्सप्लेन नहीं करना पड़ेगा दोबारा. 3 areas you need to remember: mouth, stomach, small intestine. In the mouth, the pH is 7. That is neutral. And what is the enzyme present in the mouth? Salivary amylase. ठीक है? Salivary amylase is the enzyme. And who is the substrate here? The starch. What is the pH? 7. It means amylase has an optimum pH 7. If it's at 9, it will not work. If it is 2, it will not work. Okay? Okay. In the stomach, the enzyme is pepsin, which acts on the protein, and it will only act if the pH is acidic, 1 to 2. Okay? And then in the small intestine, the pH is alkaline, 8 to 10. Enzyme. There are different type of enzymes present which are mostly working at the alkaline solution or alkaline pH. Trypsin, pancreatic amylase and lipase. Usually in तीन में से बड़ा सवाल आता है. Trypsin, pancreatic amylase or lipase. They are the one who are present in the small intestine. They work at the alkaline pH. ठीक है? Stomach works in the acidic pH. Mouth works at a neutral pH. Okay, these are 3 different areas in your elementary canal where the pH differs. Okay, and how it you can portray or how the graph comes in the paper. See, can you identify the enzyme just looking at the pH? Can you identify the enzyme by just looking at the pH?
Yes, we can
Yes.
because every enzyme has a different pH optimum level, right?
Very good. Like if the pH is 2, it means there's only one enzyme who works at acidic condition, pepsin. What is the location of the pepsin? Stomach. What is the substrate of the pepsin? Protein. देखो ये last page पे यही लिखा हुआ है. ठीक है? If the graph is at 7, ये 7 पे देखो, इसकी पीक देखो यहाँ पे 7 पे. ठीक है? 7 पे. तो 7 पे कौन है? Salivary amylase. Where can you find it? In the mouth. What is the substrate of the salivary amylase? Starch. Then if it's at 8, it's 9. Can you see? The peak is at 9. 9 is alkaline. Who works in alkaline? Trypsin or lipase. Okay? Where it is located in the small intestine. So by looking at the pH, the peak of the pH, okay? The peak of the pH, it can tell you the pH, and through the pH you can recognize which enzyme would be there, what will be its substrate, and what is the location in the elementary canal. Please remember, if they're asking about human, ये सिर्फ graph जो आपके पास given है ना, ये human के लिए. ठीक है? Examiner आपसे कोई भी condition दे सकता है. हो सकता है कि वो बैक्टीरिया का दे दे, लेकिन वो क्वेश्चन के अंदर इंफॉर्मेशन जरूर देगा. एनीथिंग विच इज नॉट नोन टू यू, ठीक है, बट यू हैव अ कॉन्सेप्ट, एग्जामिनर ऑलवेज गिव्स द इंफॉर्मेशन इन द क्वेश्चन. ठीक है, सो यू कैन टेक आउट द क्लू एंड यूटिलाइजेस इट. So that's why I say, please read the question. Please read the question because the question has the clues in it. Question has the cracks in it. ठीक है? If you pick up the crack or the code, you will easily interpret the question. ठीक है? Find out the code so you can crack it. Crack करना है आपने code को. Question जो है वो आपका question में ही clues होते हैं. You just need to find out the clues. Okay? फिर हमारी class end हो रही है, ठीक है? You can rejoin. Then we'll start our last chapter. The next chapter, that is human nutrition. The major one. ठीक है? Okay.